session here, who's Matteo Chincarini, and he's with the University of Teramo in Italy. I have to admit I had to look up where exactly Teramo is. I'm sorry. Matteo is a research associate, and his research interests are farm animal behavior, also linked to farm animal health and welfare. And judging from his publication list, he also really works with various species. And I'm really looking forward to hearing which species you are going to talk about today. And looking forward to seeing you and hearing your presentation, Matteo. The floor is yours.
Okay, thank you very much for the introduction. And I was trying to understand how to share the screen,
there should be a button
yeah,
below on this Zoom window, share screen.
I don't understand.
The green one.
View screen, and let's see. Basic. Okay, I think yes, Zoom is asking me to give the permission, but of course. Sorry. Let's try. If you do you see the screen?
Right now, I don't see your share screen.
Because the Zoom told me that maybe I had to log in again. Let me try one more time. Maybe it should work.
Yeah. Yes, it works.
It works?
Yes, I see your screen now.
Okay. So thank you very much for inviting me, and I'm very happy to be here. And so the title, yes, of my talk is looking to the sheep brain, the use of fNIRS and its limits. But when I was speaking with Christian, when you started to organize this meeting, we were discussing also about the cognitive abilities of the animal. And so I know how to speak about fNIRS, but I was trying also to fit the topic of fNIRS on the topic of cognition. And okay, so this is an overview of my talk, and I will try to make a framework of what I mean with cognition, and try to convince you why we should look into the brain also when we work with the cognition, and how to do that. One of the possibility, of course, the example of today, it will be the use of fNIRS technique, and then we conclude with some more work that we can do and a take-home message. And to be honest, I have to say thanks again to Rebecca, because she already introduced it very well, the concept of taking more parts of the problem together in assessing the behavior and the welfare. But when I started to look at the definition of cognition, I found mainly two I think very agreed definition of it. One, I think we are very familiar with the definition from Satterword. So the cognition refers to mechanism by which animals acquire, process, and store and act on information. More recently, Lyon and colleagues specify the adaptative value of cognition for an organism. So this because this definition is not only a mechanism like for respiration that imply mechanism for acquiring, processing and acting on information, but is to have a mechanism that has an adaptative value. And what for me is interesting from these two definitions is the use of the word mechanism. So what do we mean with mechanism? I mean mechanism for me is a system of parts working together. So we have different parts. Let's see an easy example of a sensory mechanism. So like vision. In vision, as you can see, we have the visual field, we have the eyes, the anatomical part, then we have the connectivity of these parts. So we have the external world, internal world, and I'm not going to add other issues of this problem, like we see that the information is going through the anatomical fibers and to the primary visual cortex. Then we could argue and discuss about how this information is perceived from a consciousness point of view. And this is just to give you an idea about of all the parts that put together make the mechanism of vision. But today we are speaking about mechanism of cognitive processes. So which parts of this mechanism can be considered? Well, someone is saying that cognitive performance of an individual are a function of structural function combined with structural and functional combination. So we have the anatomy, but also the connection of the information. And McBride and Morton defined some markers for cognitive capacity based on neuroanatomical attributes. So for instance, here we have endocrinal volume, girification, and I will stress a little bit the number of cortical neurons as an information for the measure of the connectivity of the cortex. So these are part of the mechanism of the cognition. And I said, looking only at the behavior sometimes can mean studying the epiphenomena of the mechanism that here is represented by the Gran Sasso mountain from Teramo in the center of Italy. So we need to understand all the parts, I think, that we want to if you want to understand fully this kind of mechanism and how and measuring both neuroanatomical attributes and cognitive performance, so behavior and neuroanatomy, we can have a measure of the cognitive capacity of the animal. Someone also is arguing that brain measure can explain the nature of the magnitude of many effects on welfare. I think in this case, we are looking at the brain modification in the long term, as we have discussed it also earlier. So how we can look, which kind of information we can have from the neuroanatomy. So really, we have a lot of many techniques, depending on the aim of the information that we want to use, and depending also on the information that is based the technique. So for instance, we have electric and magnetic signals, EEG, MEG, structural studies, so but also inside each technique, we have more application, like the pictures that you can see here is a special case of electrophysiology, so something related to EEG, but in this case, we are speaking about microelectrocorticography. And you can see we have also neural markers of activity. So depending of the technique, we have more invasive or less invasive way. In this case, we need the tissue of the animal. So it's something that you can detect only when the animal is dead. And I didn't add here another information that could be related to the plasticity of the brain. And we could speak about the immature neurons in the cortex. This is something also quite new. And today I'm going to speak of a special case of this technique, that is the fNIRS, that is based on the hemodynamic signal, like for the functional MRI. So how does it work? Basically, it works with light. This is a schematic representation of a sensor where you can see that the light source and light detector have a fixed distance. And when there is some brain activity, you usually have an increase of oxygen consumption because neurons need oxygen to work. And this increase on oxygenation is accompanied by an increase of cerebral blood flow. And this become what we call the neurovascular coupling. So that leads a change in local concentration of oxyhemoglobin, the red line, and a decrease of the concentration of deoxyhemoglobin. So we need to have these two lines, let's say, in the graph that happen at the same time to speak about a neural activation in the brain. So the light is absorbed by this chromophorus, the oxyhemoglobin and the oxyhemoglobin. So depending on the change in the concentration, the absorption will be different. This technique is quite useful because we can apply this technique on organisms that are free-moving, so free-moving subjects. But before to go further, I want just to highlight some characteristic of this signal. So the signal is a combination of different hemodynamic characteristic. And we also have neurovascular coupling coming from a systemic interference. So like we are breathing, our heart rate. And also it is important to understand the source of the signal. The source of the signal can be intracerebral, that is what we want to study. So what is happening in the cortex. And also we have signal coming from the extracerebral. So we need to distinguish these two parts of the problem. I didn't mention that with this technique, we can reach mainly the cortex area because we cannot go very deep in the reaching the signal, the hemodynamic signal. The depth that we can reach depends mainly on the distance that we have from the light source and light detector. Let's say more or less that we can reach half of the distance of these 2 points in depth. And now the question is it possible to use this in animals? Well, I was working mainly on sheep, so I was wondering about sheep, and the answer was yes. The group led by Lawrence Gigatz has shown that it was possible to apply this technique in sheep. We have also other studies on other species. Jan has worked with goats, and this also was another more recent publication on sheep. So we use it. This is how it looks like, the sensor. I think it was interesting to you to see how it is physically. And you need to shape the head of the animal because the air can affect the quality of the signal. And so you can apply this device and see which is the brain activation during some, let's say, behavioral task. But if we look at some cognitive abilities, and let's speak, for instance, about the learning, so the capacity to predict what is likely to happen next based on an early stimuli perception. We train the sheep to anticipate something that we were considering positive for them, that was a food reward. This study took a lot of time mainly to habituate the sheep to be separated from the flock. We know that they are social animals, so it's very stressful for them to be alone. Then you have to habituate them to wear the device. They get used to it quite quickly because it's very light, it's not annoying. And the sheep went to a classical conditioning to anticipate a sound, water bubble sound, natural sound, and then the sheep were deterring this obstacle to, after hearing the sounds, to reach the food reward. And we had to increase the anticipatory phase because when you want to use fNIRS data, you need at least 7-8 seconds of delay to record the hemodynamic response. And quickly, I show you the results of this. Here, wait, I'm sorry. My screen is going to switch off. So up on the left, you see the individual signals. And usually when you use this technique, you analyze these signals with a group average hemodynamic response. And We were also looking at the different brain hemispheres activation. So in general, there was a neural activation. You see the increase of oxyhemoglobin and decrease of the oxyhemoglobin. And the right hemisphere was more involved in this process of anticipation. At the same time, we were also looking at the behavioral reaction of the animals. So the first thing that we thought was that we had, based on the emotional balance hypothesis, a right hemisphere that was more dominant, and this could be related to some negative emotional states like frustration. But we were expecting to see something different because they were anticipating something that for us was positive, a food reward. But also alternative hypothesis could be that the sheep in that moment was mainly involved in attention processes. And when we looked at the others publication, because we wanted to compare our data, we found some discrepancies. So, I mean, we had from some case, the behavioral data were according to other literature, but the fNIRS data were not in agreement with the other studies. So something was mismatching. So we made a step back and said, okay, let's see what are we recording exactly. And just to give you an idea, as also Rebecca stressed before, if you type on Scopus brain and human, you will see almost 1 million and more documents from the 30s. If you look at the animals that are more used in the, let's say, more classical lab animals, mice and rats, we have more than 800,000 documents, almost in the same range of years. But when you go to sheep, and I had to start to count from the 1922, you see the difference. So this number means the difference in the kind of information that we have today about the farm animals. I think that is similar results we can have also with other farmed species. In sheep, we found this interesting review about the cortex area and activation of these areas. But as you can see from these pictures, the studies were quite old, and in general, there is a quite good agreement between people that are studying the cortex activity about the localization of the motor area. But we have many areas where we don't really know what is going on in the sheep at the moment. Okay, and we wanted to put a device on the head of the sheep and study that. So that was a problem at the beginning. So what we did another study where we defined with a more precise way where we were in the head of the animal. So first, from the MRI, we defined the distance, the thickness of the head, and the positioning of our sensor. So this point represents where we were with the sensor light sources. And with our colleagues, they made another neuroanatomical study, and they defined that this area where I'm pointing now is the motor cortex area, and these are the descending motor tracts fibers. This is a DTI images. So we were sure that the light source and detectors were positioned on the motor cortex area from a neuroanatomical point of view. And also, if you remember, I was telling you that the source of the signal can come from extracerebral and intracerebral tissues. So we had to do something different, and this with the help of the colleagues from Polytechnic of Milan. And we had a shorter distance to analyze only the, let's call it the scalp hemodynamic change, and and the long distance where we were sure to reach the cortex. So we had this two model level of study, the fNIRS signals. It is a more complex model that because it was needed to disentangle the superficial signals from the cortical signal. Something more that we needed to add, I didn't mention before, is the differential path length factor, DPF. DPF in human studies is well known to change with age in people. So this is a correction factor that you need to take in account because yes, the tissue are considered transparent, but the optical properties of the tissues change with the age. And in animals, still, like for the neuroanatomical information, we don't have many information of it. We didn't have. Actually, we had something just from the that anymore. So we had also this information. I'm going quickly because I'm running out of time, sorry. We repeated another experiment with this new setting of the device. So we had sheep working. We trained the sheep to work very calmly for 30 seconds and then to wait for 30 seconds. And we were recording the brain activity. And also we performed a startling test. So we were opening an umbrella. The sheep didn't see an umbrella before. This is a sheep wearing the device. And we were looking at the reaction after the opening of the umbrella. So a fear reaction. These are the results. So as you can see, when sheep were walking, there was a very clear neural activation in both hemispheres, right and left. Here we are in the cortex. This is the more superficial signal, so we don't have any signals annoying our recording. And this was what happened when we performed the fear reaction test. And we divided the sheep in the kind of the reaction that they had. So we were recording the behavior of the sheep, and some sheep were reacting, moving, so because they were afraid. Some were freezing, so they were still. And the difference that we found in the hemodynamic response was that sheep that were moving showed neural activation like in the motor task, while the sheep that were not moving after the fear, after the umbrella, didn't have any change and any neural activation. So that's to say that the application of fNIRS with this setting, short distance, long distance, multi-layer approach, can be used to, I would say, to study more motor tasks. So this could be very useful for motor tasks. For other kind of task, I don't know, we should discuss about that. And this is part of the take-home message. So cognitive and maybe emotional processes need to be approached with neuroanatomical attributes and cognitive performance, so to look at the behavior. One problem that we have today is that we miss many neuroanatomical and functional information, especially from farm animals. We don't know what's going on. And we should select the best analysis for our goal. And about that, I am going to add something else, just to conclude. So can we do something else? Other techniques? In the introduction, I spoke about the number of cortical neurons as a measure of connectivity. Also, immature neurons, I said. These, of course, are invasive information. We need a tissue of the animal. But what if we could study the connectivity of the cortex in a non-invasive way? And what if we could look at the information without having the animal performing a task? So there is a technique that is called a resting state functional MRI that could allow us to make some question in this sense. And this could give us the advantage of acquiring knowledge of also information for animal welfare. So how this change the connectivity in the brain. I'm very lucky because at the moment I'm working at the INRA Institute in French. So I'm working with a team that is a lot of knowledge on this, and we are trying to approach also this way to look at the connectivity in the brain and how the resting state, there are many questions open. So which kind of resting state networks are present or not in animals, in the sheep, and let's see how they change. Of course, all this work needed a lot of different expertise, many disciplines, and big effort. So I was very lucky because I could collaborate with many friends, people with different backgrounds in different areas, neuroanatomical, behavioral areas, technologies areas. So here you can see all the people that worked on all this that I'm presenting today. And here you have all the reference that I cited. And if I'm not too late, I will be happy to try to respond to some question from you.
Thank you very much, Matteo, for your presentation. And yes, we do have some time for questions. And I think I know at least one person that should have questions in the auditorium, but we'll see. Up to now, I see clapping hands, and there I see raised hands by Liza. Go ahead.
Hello, thanks a lot for the talk. One question, you didn't say this specifically, but I just gathered it from the pictures you showed, that in the first experiment where you got these weird results with the right hemisphere activation, it looked like you said there that the sheep were tested alone. Yeah, so they had to be habituated to be isolated, and which obviously for any farm animal, it's not an ideal situation, and it's probably not how they usually learn, right? It's probably like usually most of the learning that's happening, even if it's somewhat individual processes, it's happening as part of a social group, and there's a huge social learning component. So when we isolate farm animals and look at their cognitive processes, I think besides the potential emotional issues and the stress to the animals, we might be missing something about the social aspects of cognition that are so important in farm animals. It looked like in your second round of experiments with the walking and the umbrella that you did manage to test them in groups while recording. And is that true? And if not, and I misunderstood that, do you think there's a potential to apply some of these methods also in group settings, or is it just impossible because of the method?
Well, thank you very much for the question. It is everything you said is right. Well, at the beginning, the choice to look at the individual animals was mainly based because we were afraid of the effect of the presence of conspecific on the brain activation. So maybe we were losing some information because of course the sheep were more interacting with other sheep, and so we wanted to be very focused on the anticipatory phase without any distraction, let's say. But this requires a lot of time to get animal user to be alone. And yes, because of that, we changed the approach. We were studying animals in group. Of course, you have to train the animals together as well. And then we had one focused sheep that was wearing the device. But at least it was in group, and that because of that, the sheep were not stressed at all. We perform other studies. They are still not published. So we perform something similar to the previous that we show today, and we are working on that. In that case, we changed the approach, and we had one focus subject where we were recording the activity while it was anticipating something, but the sheep was together with what we call companionship. So this were sheep just to make companion and to not have the focused sheep stressed too much from the procedure. As I said, this technique, I mean, there are advantages that I think one of the advantages is something that you can use in a in a farm. It's not so easy, but you can apply in animals, and you can think about different kind of behavioral task. But this is not the only part of the mechanism. You have also to understand that we need to understand which area we are recording when we are using the fNIRS.
Next question comes from Rebecca.
Yes, I'm glad you saw me. I'm apparently one of the people who doesn't have a hand to raise. Thank you for this very nice talk. I really very much, it was really cool. And I very much appreciate also all the work you have put into validating what you're looking at and figuring out which brain areas you're looking at. That is a lot of work that is so important. I wondered, you talked very briefly about the DPF that you looked into. And I wonder do we know what that's dependent upon, that it changes across age? Because apparently, is that a white matter to gray matter ratio or something? Do we know? And is it something that could be used because looking at brain development using histology is always such an incredible amount of work. Is it something that we could use to look at brain development? So to look at changes across ages, or something, or to look at changes as animals get older, or can we do something with
that? I said easy question, not difficult. So, well, DPF is something that let me say is changing with the age, but mainly one of the tissue that is becoming more transparent. I hope that no one from fNIRS world is listening, but is the bone tissue. So the optical properties of the tissues change, and mainly it is the bone that become more transparent. I want to stress this point just because this factor is another part of the mechanism. Also, when you want to extract the relative change in the concentration of these chromophores. And it is important to be very precise with this, because in humans is what they do, and they know how it changes with age. So we were determining the DPF value with a different technique of fNIRS. There are different techniques. The one that we used is called time domain techniques. Physical properties that are different from what we have used to perform the task, that is called continuous wave fNIRS. With the time domain fNIRS, we were able to determine the DPF, and then the DPF was useful to extract the right concentration of this the oxyhemoglobin and oxyhemoglobin. We also performed the DPF evaluation at different age in sheep to see how it changed. I'm not sure that this can be somehow useful to see the development of the brain. I maybe for the development, but that's my opinion. Maybe I would look more at the connectivity. There are in humans way to look at the connectivity even with fNIRS. And problem that we face with animals is also the space of work that we have on the head of the animals. So in our case, we were able to put only 4 light sources and 2 light detectors. If you look in humans literature, you will see some helmet like for EEG with many channels, and you can perform a connectivity of work because you can see more in detail different areas working at the same time.
That would be very cool.
Yes, this is what I always say to our colleagues, the engineers, and I'm trying to stress them to say, let's do something like that in animals. It would be very interesting, but there are physical issues. So.
Okay, thank you.
Thanks. And one final question coming from Jan.
Hi, Matteo. Thanks for this very exciting talk. In that time when we did the first studies in goats together with Lawrence, by the way, I'm not expert regarding the technique, but during that time I've learned that we need, or other way around, any movement during measurement disturbs the results. So in your second approach, you trained the animals to walk along an alley, and then you confronted them with an umbrella. So I can guess that there's a lot of movement in the animal during your measurements. Was it just because our technology at that time was very prototype-like, and you have better technology now, and movements do not disturb the measurements so much anymore?
So just to clarify, our first study was made with the same device that you know.
Okay.
Well, what the problem with the movement is mainly at the head. If you look at the head, this is something that you have to remove. You will see also from the signals that the signal is very noisy, so it is quite easy to detect. The prototype that was developed from Martin Wolf also had an accelerometer, so you could see the movement of the head, and this is something that you don't want because this is affecting the signal for sure. Not a stable movement, like if you walk calm in a calm way, let's say, this doesn't affect the signal. But if you walk and you start to shake your head, yes. So this is something that you need to do either with an accelerometer, or if you have a camera, you can see when the animal is moving too much the head, and then you have to avoid this kind of information. Because of that, it was necessary to train the animal to wear the device, to be calm and not stressed. And we were removing all the signal that was too noisy because of the head, because that could be interpreted like an artifact. I mean, if you think about, if you look also in human literature, that is still recent, but of course there are more studies on that. There are many tasks with a different fNIRS device weared by people that are cycling or running, and you can get a good quality of signal. The problem is if you move a lot the head.
Thanks.
Yes, thank you very much for this excellent presentation and discussion, Matteo. And thank you to all the speakers of this morning session and everyone who contributed to the discussions. I think it was a very interesting morning already. And now I will hand over to Jan, who will have some organizational stuff, I think, about